I guess. Let's start on time. This is Tuesday seminar series of Institute of Biomedical Engineering, organized jointly by Istanbul Health Industry Cluster and Center for Life Sciences and Technology of Boğazici University. Today, we have a very interesting topic and a very distinguished guest, researcher, İbrahim Tarık Özbolat. He is an associate professor of engineering science and mechanics at Biomedical Engineering and Neurosurgery. Also a member of the Huck Institute of Life Sciences at Penn State University. Dr. Özbolat's main area of research is 3D bioprinting. Unless he managed to print a copy of himself, he is in person, connected from a very nice place somewhere in Turkey, which he may or may not disclose to us. It's a great pleasure to have him as a guest speaker to our seminar series. The floor is yours, Dr. Özbolat. Thank you for coming.
Well, thank you very much for the invitation. So it's a great privilege for me to be here with you guys today. So I'm joining from Adana, Turkey. So it's not really a super private information, but I just wanted to share with the guest. So today I'll present our research on 3D bioprinting, and this is particularly for fabrication of tissues that can be used for regenerative medicine purposes, for tissue engineering, as well as disease modeling. I have been in the field of bioprinting for about 13 years. So what we do in the lab is quite interesting, as we use 3D printers to manufacture tissues. So it's I mean, using 3D printers are very typical nowadays, even in the industry. But what we use is we replace the non-living materials with living materials like living cells, and then we print them layer by layer, and we make various different tissues like bone, cartilage, blood vessels, composite tissues, pancreas, lung, skin, and cardiac and tumor models, and even recent lung study where we build these tissues that we can use for regenerative medicine purposes, like bone regeneration, for craniofacial defect replacement, or it could be skin regeneration for skin defects, or it could be like tumor models to understand the cancer progression and metastasis, as well as recently we started some interesting work with the lung tissue engineering. Since we haven't really published this yet, I'm not going to disclose more information, much information about the lung, but the goal with the lung is to create a lung model at the alveolar space that can be used for understanding different diseases, even like recent COVID-19. So today, here is the agenda for my talk. So I'll start with bioprinting. So I'll just give a very brief introduction about bioprinting. Then I'll talk about the overview of the 3D bioprinting work going on in my lab in the last 10 years, since 2011, when I started my lab at the University of Iowa before joining Penn State. Then I'll talk about the aspiration-assisted bioprinting technique, which is a new technique for precise positioning of tissue building blocks. And then finally, I'll talk about the intraoperative tissue printing, so printing things directly on a defect in a surgical setting. So a couple of years ago, I was invited for a talk at Boğazici University. So I talk about the intraoperative tissue bioprinting work in details there, but I just briefly discuss our recent progress with this technique. So here is an animation of bioprinting. So this is not real, it's just the animation. So what we do with the bioprinting is we put the living cells in the cartridges of a 3D printer, and then we deposit the living cells in materials like hydrogels. Those materials are biomaterials, and then we deposit the cells layer by layer precisely, and then we build a structure, and then over time we expect that the structure, particularly the cells, they basically organize, and then they remodel the tissue, and then eventually we get a tissue that could be readily available for transplantation. So what you see there is a heart that's being printed with vasculature, multiple cell types, and in the shape of the real heart of a patient that could be readily available for transplantation. But we're not there yet. So this is still ongoing. We have made significant progress, not just our lab, but the Bioprinting Society in general. We made quite a bit progress in the last 10 years. So 10 years ago, we could be able to print living cells. Now we can make tissues, even like some anatomically correct organs, but not really at the biological or functional level. Hopefully in the next 10 years, we'll make things like, organs, solid organs like heart, kidney, liver, and we can use them for different purposes, such as, transplantation. So in the lab, I'd like to talk about some technology that has been around for 10, 15 years, where we use melt extrusion technique, and then we 3D print scaffolds like this that are porous, and then we use different polymers, and particularly we use a composite polymer made of PCl, PLJ, and HA that's biodegradable and biocompatible. So then after printing, we loaded that on red calvarial defects. So as you can see here, the micro CT scans. So after implantation, we see that we got some bone regeneration with the composite material that we use, but even like that, material induces some vascularization, as you see some blood vessel formation, and in general, we got better results with respect to just empty defect. This is a critical size defect where the defect itself can't really repair itself without any intervention. So the PCl printed group, PCl was a control group where PCl is a clinically available material. It has been widely used for bone regeneration purposes. And then with the addition of the composite material, we illustrated better bone regeneration, but it was not really super satisfactory. So then we further improved that, and then we integrated the gene editing technologies, the gene therapy technologies, where we use the microRNA 148B, and then we transfected the bone marrow stem cells that we derive from the rats, and then we loaded those cells into collagen, which is a biomaterial, organic biomaterial, and then we injected that into printed structure, and then we implanted that, and then after implantation, we see that we could be able to pretty much completely repair the calvarial defects in about 8 weeks. And as you can see here, we could be able to breach the defect. So this was, of course, integration of the 3D printing, not bioprinting, with the gene therapy, and then now we're working on bioprinting, even like intraoperative bioprinting, where we are trying to repair these defects directly in surgical settings, where we directly print into the animal. So we also work on 3D printing techniques that can be used for lab on a chip or organ on chip applications. In this regard, PDMS is one of the popular materials used in organ on or tissue on chip devices. We could be able to print that. Normally, this material is not really printable, but with using different copolymers of PDMS, we could be able to tune the viscosity as well as the rheological properties, and we could be able to print that. We could be able to make various complicated shapes. The interesting thing with this material is with the 3D printing, we demonstrated that the cells could better adhere normally on the flat surface, we can't really have a lot of cells adhering, but when we printed them, the cells actually grows on the surface better. And then we also demonstrated that the mechanical properties of the printed constructs, they were better than the cast samples. So we use the PDMS printing technology, and then we are making perfusible tissue on chip devices, like a vessel on a chip device, where we can line up the endothelial cells in these vessels and perfuse. And recently we make even like more complicated channels using 3D printing of some fugitive or sacrificial temporary materials. Once we print these materials, as you can see here, we can make very complicated arc shapes in 3D using this technology. So these are going to be generating the templates for blood vessel, and then we can perfuse these with the cell media. We can seed multiple different cell types into these structures, and we can make, various different configurations for different organ on a chip devices. In other recent work, we demonstrated the bioink development. So the bioink is the material that is used to load the living cells in it. So it's a biocompatible material, and most of the time it's a hydrogel-based material where we can synthesize these materials. For example, in a recent work, we synthesized a new material that can cross-link with the material in the support bed. So this support bed here, it is not really just supporting the printed structure, but it's also facilitating the cross-linking, like the solidification of the printed material with the cells. So you can't really see it now because it's not really super transparent, but the red material that is being ejected is being written now, is being deposited and then printed in 3D. So now you can better see. But at the end, what we get is we got various different complex structures like these cages, cages with the ball, where the ball is rotating freely inside this cage. We make complex structures like the bone model, where we can anatomically print the right shape base on a 3D model. We can even put multiple cell types and grow these cells. So this technique is also called the embedded bioprinting, which is enabling bioprinting of very complicated structures, which is not really possible using standard approaches. So in the past, we also work on some interesting concepts. So previously, we were putting cells in hydrogels, and then we were printing them. But we could also make things in a scaffold-free manner. So scaffold-free means there's no really a biomaterial, exogenous biomaterial at the beginning. So the cells are not really just loaded in hydrogel. Rather, we make little tiny capsules, and these capsules are like about like a millimeter thick. So these capsules are semi-permeable. That means the water can get in, oxygen can get into, and the waste produced by cells can get out. So then we injected the cell pellet inside. So it's made of cells only. And the cells over time, they produce their own extracellular matrix and deposit their own proteins. And over time, it makes a tissue inside this capsule. And then we dissolve the capsule by using a chemical enzyme. And then we generated the tissue strands. So the tissue strands are little tiny filaments. They are like a mini sausage. So it's the mini or micro or sub-millimeter or micro form of sausages that is made of cells. So if you make this using, say, muscle cells from cow, like the bovine, it could be a meat that you can eat, but this is pretty expensive. As you can see here, this is a tissue strand that is longer than a centimeter. So we use this for bioprinting purposes. So the goal is to load this into the printers and replacing a plastic in 3D printers, and using these as building blocks for scaling larger structures. So, but one of the problems with these strands were they were fully dense, like they were extremely dense in cells, so that the oxygen penetration was limited. So in this regard, we introduced a concept where we generated micropores inside these tissue strands. So as you can see in the MRI image, you can see the pores. So with the induction of pores, we could be able to get better cell viability. So we then differentiated the cells. We actually used the adipose-derived stem cells here. Then we inducted the osteogenic and chondrogenic differentiation of these tissues. So we make cartilage and bone out of the fat cells we obtain from clinics. So these are from the liposuction surgery. And then with the induction of these pores, we better differentiate these strands into cartilage and bone strands. But with the use of the solid strands without any pores, we show that we could be able to make cartilage that looks very close to the native tissue. As you can see here, the SK content, which is the main protein of cartilage, is very similar to the native cartilage tissue. Compared to 2D culture cells, the expression of the cartilage-specific markers like SOX9, collagen 2A, ACAN. So we could be able to express these genes much better compared to 2D counterparts. So then we demonstrated their self-adhesion, so self-healing. So when we put these two strands, they were here, the green and red, we actually transfected the cells, two different cell types, like same cell type, but with two different lentivirus, and then we could be able to fuse these two strands when we put them together. So this is showing that, say, the red cells are moving to the green zone, and green cells are moving to the red zone, and at the boundary, you see that the boundary becomes extremely loose, and over time, you can't really distinguish the boundary over time. So this is pretty much what happens in the body. If you have, say, a skin cut, a deep skin cut, what happens is the doctor puts sutures so that the skin from the both ends, right, they adhere, and the cells they cross migrate to the other side, and this is how the adhesion realizes. So this is pretty much similar to what we try to accomplish in this work. So we try to mimic how the tissue heals in the body. So then we loaded these strands into custom-made bioprinters. So we made cartilages. We also 3D printed these cartilages, like porous cartilage or solid cartilage, that we put the strand inside, and then we 3D print those strands. So as you can see here, when we print the cartilage strands, and over time, in 3 weeks, the strands, they fully fuse, they adhere, and then they make a single patch of cartilage, which was extremely intact. And when we look at the histology, it was looking like the native tissue. So using these tissue strands, we further advanced it recently, became the cover image of the Advanced Healthcare Materials latest issue, one of the latest issues. So we printed these tissue strands made of the adipose-derived stem cells that we obtained from clinics. So we differentiated them chondrogenically, and then we made tissue strands, and then we printed them using an interesting concept. So this is, I'm going to explain that concept later on using the aspiration-assisted bioprinting technique. But the interesting thing that we observe with these tissue strands is, as you can see here, when we look at the ultramorphology of these strands, we see that the collagen orientation was longitudinally, the collagen fibers, they were longitudinally oriented with respect to the strand axis. So as we know that in cartilage, the superficial zone on the very top part of the cartilage, right, the collagen fibers are they are horizontally oriented. When you go deeper, close to the subchondral bone, like in the deep zone of cartilage, the fibers are vertically oriented. So by mimicking this technique, we actually fabricated the cartilage tissue, where we use these tissue strands. Here, the tissue strands on the top zone, they were horizontal oriented. In the bottom zone, they were vertical oriented. And then we look at the ultramorphology, and then we show that by controlling the strand orientation during bioprinting process, we could be able to control the collagen deposition. So by using these tissue strands, we also show some other interesting concept in the past, where we wanted to 3D print the tissue strands with the vasculature. So the vasculature is essential for fabrication of scalable organs. So in this regard, we use a multi-arm bioprinter that we developed back at the University of Iowa. So using this hybrid approach, we demonstrated that we could be able to make perfusible tissues. But just want to give a couple more information about this vasculature, particularly coaxial bioprinting technique that we use at the time. So with the coaxial bioprinting technique, where we have two nozzles, one nozzle is embedded inside the other one concentrically aligned, so that we can bioprint the material inside the outer section, and then we can bioprint or extrude or dispense the crosslinker inside the core section, so that when the crosslinker contacts the material, it crosslinks immediately, and then we could be able to get some tubular structures right away. So this was highlighted in Popular Science back in 2013. So with this technique, we could be able to bioprint the vascular network regardless of its length. So for example, the middle image shows like about a millimeter long structure. We could be able to make branch ones by manually suturing some stem part of the other vessel. So when we culture them over time, we could be able to grow cells, and particularly we printed these vascular conduits or vascular network within larger scale hydrogels, and then we could be able to show that with the integration of these vascular network, we could be better sustain cell viability over time. So this technique has been adopted by several labs nowadays in the world. I can say at least 20 different groups are using this technique for fabrication of vascular network. So then we combine that with the tissue strand printing. This was an interesting concept where we print these tissue strands. So as you remember, the tissue strands they have the self-healing or fusion capability. So we printed these with the vasculature in tandem, and then over time we expect that in a couple of days these tissue strands, they self-assemble, and then they enclose the vasculature in full. So this is the experimental image where we use a support structure. This is just to support the entire structure until fusion completes. So after printing, we see that we could be able to make a tissue that is enclosing. This is a stromal tissue that is enclosing a vasculature. And this could be used for various different purposes, like could be used for disease modeling, or even could be used for transplantation. So in another study, we use the bioprinting work to create pancreatic organs or even tumor models. So with the pancreas, it could be for multiple purposes, for making a transplantable structure, a transplantable organ that can be used for treating type 1 diabetes. In the meantime, could be used as a little platform technology to detect the response of different type 1 or type 2 related drugs. So here, of course, the vascularization is one of the main component, as vascularization is really important. So we show that we could make the pancreatic islets using different cells, such as the rodent cells, and we combine them with the endothelial cells. We're now doing that from IPS, the induced pluripotent stem cell drive beta cells too. So in the published work back in 2018, we show that we make these pancreatic islets. These are engineered islets. And then when we look at these in details, we could detect the insulin granules. So even we made these with the cardiac cells, so the stem cell-derived cardiomyocytes, and then when we put them in a gel, we see that these contracts, since the gel is pretty stiff, the contraction was slower, but when you put that outside it, so the contraction was faster. So with the pancreatic islets, we showed that we could induce vascularization, intravascularization. So as you see here from the islets, we can sprout out capillaries. So with the capillaries, we improve the viability of cells, beta cells, as well as we improve the insulin production function of these beta cells. So induction of vascularization is not just important for the viability of the insulin producing beta cells, but also improves their insulin production function. So then we recently completed a work that is about to be submitted in the next couple of days. So in this work, we basically create a 3D model of a cancer microenvironment. So using a similar concept where we can sprout out capillaries of the tumors that we created in three-dimensional hydrogels, and we even show that these capillaries are here. So the cancer cells can invade into these capillaries, and then this system can be used for mimicking the cancer growth and metastasis processes. So in a collaborative work with Darya Unutmaz, we also show that we can engineer these system. So basically, we use the immune cells, and these immune cells, the T cells, they can recognize the cancer cells. So we engineer these immune cells to recognize these cancer cells, and then can attack the cancer cell immediately and kill them. For example, these are some engineered immune cells. So here's the cancer cells. This is just an ACM image showing the attacking of these. And then as you can see here, the cancer cells spheroid here is being attacked by the T cells. So here, the blue dots, they show the immune cells attacking cancer cells. In about like a couple of days, 2 days, we could be able to kill the 90% of the tumor. Of course, this is a relatively simple model. So our goal is to make more complicated models using bioprinting technology, where we can precisely locate these tumor spheroids, and then we can grow vascular network that we can perfuse the T cells as what happens in vivo conditions, right? So the cancer cells, when they grow, right, over time, you expect that if it's highly metastatic, then grow and metastasize into other organs. So then these blood vessels are grown by the tumor environment, and then over time, we expect that the cancer cells, they can intravasate into these blood vessels, and then we can perfuse the engineered T cells, and we can explore the interactions in this platform technology. So we have made some progress. So for example, here in a recent work that we completed, we could be able to create these tumor models and put them, and then we have these vasculature. And as you can see here, the vasculature can sprout out capillaries and that are anastomosing to the main vasculature that we created, and then we're still trying to improve this, and so that we can perfuse T cells and explore how these engineered T cells interact with the tumor. So in order to print these spheroids or these the tumors, we created a new technology called aspiration-assisted bioprinting, and we published that about 6-7 months ago. So in this technique, we use the aspiration forces to aspirate the spheroids, to lift the spheroids and mini tissues, and then we can precisely print them. For example, here, this animation shows the aspiration of little tissue spheroids, like they're balls, and then we aspirate them, and then we can lift, and then we can position them onto hydrogels, and these hydrogels could be sacrificial hydrogels. It's just a support hydrogel where we can put these balls, and then we can cover that by another layer of, overlay that with another layer of support support material, and then we can later decrossing this support material, dissolve it, and then we can expect that these spheroids can fuse, so we can make large scale tissues by using the same principle that we did with the tissue strands. So these are tissues that when you put them together, they fuse, self-assemble, make a larger scale tissue. Then we also show that we can actually print these into functional hydrogels, like fibrin, for example. So fibrin is made of fibrinogen and thrombin. So when they interact, they cross-link. And when we put these spheroids, we can precisely print them in 3D, not really in fully in 100% freedom, but we have some freedom at least at the superficial layer of the gel. So then these spheroids can sprout out capillaries, and we can use them for different studies. So we first explore the physics of that. I'm not going to go into details about the physics, but we basically try to understand how the tissue interacts during the lifting process. And then after understanding this, then we try different spheroids made of various different cells, like HUVECs, 3T3 fibroblast cells, different cancer cells, even like the combination of multiple different cells like MSCs and HUVECs. And we see that as we have the compaction increases, as you can see here, these are like more compact spheroids, as the surface roughness is less compared to the spheroids, like HUVEC spheroids. Then the mechanical properties of the spheroids increases, like the surface tension. So we look at the bioprinting parameters that are very critical to precisely aspirate them without breaking them. So in this regard, we measure the lifting pressure, and then we predict that with some mathematical prediction tools. And then we also look at the deposition of these spheroids onto a substrate. For example, when you deposit that, it should stick. And then we also formulated that, and then we optimize our parameters to get these spheroids sticking on a substrate. Then we demonstrated that we can print these spheroids into various different hydrogels. Like we can make, different patterns, like, lines. We can make triangles. We can make very complicated shapes using, again, these are just 2D, by the way. So these are on a substrate, on a surface, so we can make different spheroids at different sizes and shapes and types printed on a substrate. So they don't have to be a spheroid, by the way. As you can see, there are some irregular shapes. So we try printing on JALMA, we try printing on collagen, different hydrogels. We also try to print those on top of each other in the air. So this was an interesting video that the students captured. As you can see, like 8 spheroids were printed on top without any support structure. This was very interesting. It's like a pizza tower, right? It's not really tipping off, and this was extremely interesting, got a lot of attention. So we made different configurations like bridges and so on. So we also demonstrated that we can use this technique for understanding the angiogenic sprouting of capillaries. So how the blood vessels are sprouting from capillaries and how they interact if you put 2 spheroids next to each other. And then interestingly, we show that once we put 2 spheroids next to each other, then if there is a neighbor spheroid, for example, the green and the red spheroid, since the red spheroid is on the right side of the green spheroid, so here the sprouting of capillaries from the green are affected by the sprouting of capillaries from the red. So there is some sort of a directionality in between. And then we printed 3D configurations like this pyramid shape. As you can see here, these spheroids, they don't have to be the same size. So it could be, different size. The green ones are made of different cell types. So they're in the middle. They're the mesenchymal stem cell spheroid, and the red ones are HUVEC. So then we recently published another work that we show the fabrication of osteogenic spheroids, like bone spheroids, like the mini bone tissue. So we make these bone spheroids and we printed them, and when we show that with the 3D culture, and particularly with the inclusion of HUVECs, these are the endothelial cells, we demonstrated better differentiation of these MSCs towards the the osteoblasts, like osteogenic cells. So then we printed various different configurations where we can exemplify this further. So I have a couple more slides that I couldn't really show as we haven't really published yet. But as you can see here, we can make these various complicated structures like different 3D shapes, and then and when we micro CT scan them, we could be able to demonstrate the deposition of the bone tissue. So we also show this technique for the osteochondral tissue printing, and particularly the combination of the cartilage and bone at the osteochondral interface. So we basically we use the adipose-derived stem cells, and these adipose-derived stem cells were obtained from clinics again. So we differentiated them osteogenic to make osteogenic cells like bone spheroids, and then chondrogenic spheroids and cartilage spheroids. And then we print them, as you can see here. We could be able to make the cartilage zone at the bottom here, and then the bone zone. So the interface was of course here. It was very clear. So this is just a platform technology. Again, we are working towards like more automation, as well as like more capabilities in building scalable structures. But recently, we demonstrated another interesting concept with the use of a support bed, and then we could be able to use the embedded bioprinting technique and combine it with aspiration-assisted bioprinting, where we can pick up the spheroids and then we can precisely print them into different locations in a support bed. As you can see here, now it's printing the PSU shape. So this was highlighted by various different media outlets like Genetic Engineering, where we can make extremely complicated structures. So we're further improving this technique, and particularly the support bed here is very critical. So some support beds are not really super nice for biological applications, and some support beds are not really super nice in enabling precise deposition of these spheroids. Sometimes you can't really precisely put that in the designated point. So in this regard, we are actually synthesizing and developing new support beds that could precisely bioprint, that enable us to precisely bioprint these spheroids, as well as being transparent. That means you can see it as you see in the video, right? Because this is very important, and biologic defined and facilitates the fusion of these spheroids. So using this technique, as you can see, we made these spiral patterns, and these spiral patterns are very, very difficult to make using conventional techniques. We can make these tubular structures, and we expect that these spheroids fuse. We actually show that they fuse, and like this double strand of a DNA shape, and different examples like a cage with a small spheroid that is enclosed in the middle. So in the last part of my talk, I'll introduce the concept of the intraoperative bioprinting. So intraoperative bioprinting is a technique where we directly print things on the animals. So as you can see here, there's a rat model. It could be mice, or it could be even like a larger animals. Like we're also doing some sheep studies nowadays, where we can create a defect in the surgical settings. We put the animal under the bioprinter. So we have a 3D scanner, and it scans the defect, and then the printer goes and then prints multiple compartments. So particularly for craniomaxillofacial defects, where we print the bone first, and on top of that we print the intermediate layers like periosteum, and then on top of that multiple layers of skin could be printed. So in a big project that we've been engaged, we are now using some interesting concepts like the gene therapy as well, where we can use the gene therapy to stratify the tissue. So just print stem cells, and then by controlling the locally delivered materials, like it could be biomaterials as well as the proteins or any genes inside or microRNA. So we could be able to control the local differentiation of these stem cells into different lineages. So the goal here is to print a bone first. That's just the calvaria, and on top of that there is a barrier. So at this point it's a barrier. It's just to eliminate the sedimentation of the cells from the dermis layer, because these are soft tissue cells, so they shouldn't really migrate down because of gravity. We want to prevent that. And then after the barrier, we print the dermis, and on top of that, the epidermis layer. So it's a 4-layer structure at this point. So we are interested in adding more to recapitulate the native tissue better. So far, we developed a material called hard tissue ink. So the hard tissue ink is a collagen-based material, and we could be able to print in air cells, the stem cells, as you can see here. The cells, they grow, they like the matrix. We have a good control on the structure, fidelity, the shape-wise. And then after demonstrating the printing, we also show that this material is osteogenic. So when we put the stem cells, we could be able to differentiate these stem cells osteogenically. They were showing positive for RUNX2 and BSP at different time points. And we also look at the genetic expression of different bone markers, and we show that just the material itself, without using any induction media, is sufficient to do osteogenic to differentiate the stem cells. So, excuse me. Then we move to the surgical settings. Unfortunately, I can't really show the surgery video, as the presentation will be publicly available. So after we drag the print the bone tissue first. So these are bone-only defects. Okay, there's no skin defect at the beginning. It's just a bone-only defect. And we printed the material, and 6 weeks after implantation, we see that we got substantial, about like 80% bone regeneration. So in one of the groups, we actually use the recombinant BMP2, which is a clinically available material, as demonstrated bone or supports bone formation in clinics. It has limitations as well, but since it's clinically available, we wanted to show its impact too. So then we also work on the skin regeneration. Of course, for skin printing work, the problem is the resolution should be high. So the main problem with the skin printing is the skin layers are very, very thin, and we can't really extrude the material because the extruded filament thickness, it was greater than the thickness of the skin layer. So because of this issue, we use the droplet-based bioprinting technique, like it's like inkjet printing technique, or it could be microvalve, where we can deposit the jets of fluid, like the bioink with the cells. So these are with the skin cells obtained from rats. And then we show that by controlling the parameters, by optimizing the parameters, we could be able to control the droplet formation. So then we intraoperative bioprinted these skin layer on a rat model. Again, it's on the craniofacial area. So these are the data that shows skin regeneration on skin-only defect. Okay, so 6 millimeter defect on the cranial area. So we didn't really have the bone group or the bone defect in this set of experiments. So we had 4 different groups, like negative control, like empty defect, or the ST ink, the soft tissue ink, or the ST ink loaded with the red dermal fibroblasts, and we have 2 layers stratification, like the dermis printed first, and then the epidermis. Epidermis was basically obtained by printing the ST ink with the keratinocyte growth factor. So after like 4 weeks, we got the explants. So when we look at the explants, we show that the printed samples were better in terms of the mechanical properties. So we did the punch-out test. We insert the needle, and then we determine the mechanical properties of the skin. And then when we look at the histology, with the printed groups, we got better skin regeneration, faster reepithelialization, as well as more collagen deposition, as you can see here in the Mason trichome images. So it was clear that the epithelial layer and then the dermis layer were regenerated in 4 weeks in full. So then we combine intraoperative bioprinting of bone with the skin together. So this is a this was very first time in the literature that shows the integration of multiple different tissues with distinct properties for the craniofacial area and reconstructed them in surgical settings where we printed the bone layer first, as you can see here, a little barrier layer again to eliminate the sedimentation or migration of these soft tissue cells or the cells from the dermis layer to the hard tissue layer. And then on top of that, we printed the skin. In about 4 weeks, the skin fully regenerates, and then we got bone regeneration. And we got substantial bone regeneration in week 6 versus week 3. But compared to bone-only defect, again, the bone regeneration was limited. This is because in the composite defect, right, the defect was open all the way up to the air, so there is limited vascularization, right? Since there is limited vascularization, bone regeneration was limited, but still, the results were extremely encouraging, as we are now trying to integrate the vascularization and other layers like the adipose layer of the hypodermis, as well as the periosteum that stays between the bone and the skin, that is actually rich with stem cells and vascularization that supports bone growth. So the next project is still we're still working on that, but we did one experiment before COVID-19 came through. So we stopped the large animal experiments, but hopefully in 2021 we're going to resume our experiment. So in this experiment, we want to regenerate the bone on large animals. Like we work on sheep, that is funded by the Osteology Foundation from Switzerland. And then our goal is to regenerate the bone tissue in surgical settings for sheep that is actually close to the human physiology. So rats are small again, but they are important to understand the outcomes of our approach. So we need to translate this into clinics. Right, the goal is to translate this technology into clinics, which is actually the cover image of my book. That is the very first book that fully covers the bioprinting, comprehensively covers the bioprinting. So here, as you can see here, when the bioprinter gets and translated into clinics, and we see that these are gonna help to replace the damaged or diseased tissues of humans. Of course, if this could happen, I could be the first person that can go under a bioprinter. So this was an interesting image that I myself went under a 3D printer in Long Beach, in California, back in 2015 in the Rapid Symposium, which is the biggest symposium in 3D printing. So I was asked to go under a 3D printer and then just sense how the intraoperative bioprinting looks like, and it was a little scary, as what I had on top of my head was extremely hot. It was a melt extrusion 3D printer for plastic printing, but at least it was a different feeling. Of course, this is a very brute attempt, so it's just you can't really use typical 3D printer to print things on the people. Of course, we are using like a robotic arms, as you can see here. They are like a robotic surgery tools like robotics surgery has been around for years, right? For a couple decades. So why not the 3D printer concept could be integrated into surgical robots, and robots can directly print things on humans in clinical settings. So with that, I would like to conclude my talk here. So I want to acknowledge all the students and visiting scholars, as well as the collaborators. Over like 50 people contributed in the last 10 years in the lab. As you can see here, this is our building at Penn State University. So we're on the third floor in this amazing building. And then, of course, without these supporting agencies, we couldn't be able to accomplish this work. So I really appreciate their support and help. So I think we can stop here.
Excellent. Thank you very much. This was an excellent talk, and it was watched very carefully here at Zoom, also in YouTube. If there are any questions from the audience at Zoom, or even I can keep an eye on the YouTube, they can ask questions now to you directly on the few minutes that we left. If we stop sharing maybe so we see each other faces a little bit better, it will be probably a good idea to stop the sharing. And I asked the students to turn their videos on if they have no big